Super. Thank you very much, Gwen. That was great. So the 6 learnings of Gwen. And Gwen works at Leuven University, and the Leuven University has been a leading group in Europe and in the world if it comes to microbiome research. And what I really like is that they bring in the physiology as well. So if you have a burning question to Gwen, then ask it now. But at the same time, we will have a small poll that was provided because we would like to know whether you are, as a participant of the ILSI session today, are actively engaged in microbiome research. And that would be nice if you could answer the poll that Michela hopefully can put forward. So are there any burning questions? So I have a question coming in, Gwen. It comes from Goose Cortman. He compliments on this nice overview that you gave. And here's a question. Would you consider relatively high richness still be beneficial in case of average stool consistency?
I would consider a very low richness or the low richness as a thing to avoid. I would consider it not healthy at all. Yeah. So normally, well, so we see definitely the highest richness in the hard stools. There is still some high richness in people with looser stool or individual with looser stools, but then there is a sudden decrease, a marked decrease in richness, and it's also associated with a marked decrease in bacterial cell counts, right? And that's when you move to this particular Bacteroides 2 enterotype. From the moment that you're there, I wouldn't consider it beneficial anymore. But in the range outside the Bacteroides 2 enterotype, I still think there is some space for well, that a variation in richness doesn't need to be bad.
Okay, thank you very much again, Gwen. That was a fantastic start to our session, and we see you later then in our panel discussion.
Okay, thank you.
Okay, so we now move on to more technical things in terms of having Susann Bellmann from the TIM company telling us what TIM is all about and where the limits, but also the benefits of in vitro methods are if it comes to microbiome research. TIM originated in the early 90s, as far as I recall, at TNO, and it's now a company that does sells these tiny machines. Well, some of them are a little bit bigger. And Susann has produced some really nice paper, particular one that I like, where in vitro digestion in the TIM system could be correlated with the in vivo glycemic response in humans. That really convinced me that TIM can deliver something of benefit to the science community. So Susann, the stage is yours. Please go ahead.
Thanks very much, Hannelore, for this introduction, and I'm glad to see you here back in this virtual conference, but also all of you who have joined the session. I'm hoping that you can hear me well and that the talk is understandable, and thanks also to the organizers for organizing this interesting session as well as the invitation to talk. So here is my declaration of interest, and I would like to directly jump into the scope of my talk. As the title already suggests, I will focus on in vitro models, which are non-cellular fluid models, and I've prepared a choice of models that, in my opinion, simulate the physiological reality as closely as possible, because that's ultimately the aim that we want to achieve by in vitro models, being as closely as possible to the reality, so that the results or the measurements that we generate are as much as possible predictive for what we see in vivo. So in my talk, I will compare some of the most relevant models in terms of how they look like, but also in terms of their readout parameters. To start with, and that's already presented in the previous talk, the only material that we have available to work with are actually stool samples or fecal materials, which originate from the distal colon. You can see here measurements that we did in our lab. That's unpublished data where we just compared the microbial composition of 7 individuals. And as you can see, it is very colorful and it is very diverse. The diversity reduces or leverages out a little bit once we pool the material. But there is another reason why that's not a problem, and that's shown here in this picture from the publication of Hüttenhauer, which actually states that the general microbiological understanding or microbial taxa vary, but nevertheless metabolic pathways and metabolites that are produced are stable within a healthy population. So having said that, it means the diversity of the starting material isn't that much big of a problem because the metabolites that we generate are comparable in a healthy population. That's illustrated in this paper here, where we wanted to know whether pooling or looking at individual microbial data does matter. So we looked at differences in metabolites, in this case short-chain fatty acids that were produced from healthy individuals from pooled samples or, as said, individual samples. And what we see is that the metabolite profile is the same from pooled versus individual data. In the other way around, and that was also briefly touched in the talk before me, if we have diseased microbiota, for example, or deviating microbiota, for example, originating from obese patients or obese individuals, this is different. So in this example here, just to look at, for example, the butyrate, the energy extraction from GOS, galacto-oligosaccharides, that were measured here in terms of the butyrate production, for example, that's different from lean people versus obese people. So that means energy extraction or metabolite formation from healthy or, deviating microbiota is different. Having said that, I would like to return back to the comparison of in vitro systems. As said, that all use stool materials as a starting compound. And now I selected the four in vitro methods that are, in my opinion, most published about and are out in space, which is the ice cream system. That's a 96 well-played rather screening platform developed by TNO in the Netherlands. Then we have another system that's called the ARCOL, an artificial colon system developed by the University of Auvergne in France. We have the SHIME simulator from the colleagues in Belgium, and we have the TIM2 system. Briefly in this slide, what do the systems represent? So we have the first one, the ice cream, is, as I mentioned, a 69 well plate, which has body temperature, which has inoculum with stool samples, which also uses substrate from small intestinal effluent matrix that has been prepared or reproduced. However, that system does not have a removal of metabolites, of microbial metabolites. I will come to that later. The next one, the ARCOL system that you see, and the second is a one-stage stirred vessel. Again, body temperature, pA, and anaerobiosis are simulated. By the way, that applies to all of the systems. In that system, we do have removal of metabolites. A dense active microbiota that also applies to all of the systems, and as said, supply of ileal medium. And here we already have a difference in advantage with regard to the ice cream system, is that the ileal medium is, of course, a substrate that keeps the microbiota alive and keeps them in a certain condition. Third one, the SHIME system, are 3 successive stirred vessels, also as for what concerns the colon part. Of course, in advance of that, it has also a stomach and a small intestinal part prior to that. Again, as said, body temperature, pH is simulated, which is important for the microbiome in certain regions of the colon. Active microbiota, and an optional removal of metabolites as well. Last, the TIM2 system differs in a way that it is rather a compartmental system versus a vessel. And in these compartments, there's a sort of a peristalsis applied that allows the pumping around of fecal material. Again, anaerobic body temperature, pH simulated, and removal of metabolites, active microbiota, and a continuous feeding of ileal effluent medium. Now, as I mentioned in the beginning, we have different input material, which is all the time based on fecal samples or stool samples. And I also said that the outcome of metabolites, or the metabolites that are produced, are comparable despite the various input material. And therefore, my next slide looks at what outcome parameters are we measuring, or what are the data that we generate with the system. I try to compare in this table data that are all originating from control experiments, so no intervention other than the standard ileal medium added, and after the incubation period. So the data that we have available from humans is very scarce, and so that's simply because we cannot measure short-chain fatty acids in the lumen of the colon, because it's continuously removed and absorbed. So the data that we have is, or that the literature that I found, perhaps there is more out, as Hannelore said, there is daily many publications coming out. But the data that we have here of bacterial density is based on sudden death individuals and was published with 10 to the 12th. That is followed by luminal short-chain fatty acids concentrations in human by approximately 123, but also that varies depending on diet and status and so on, and a short-chain fatty acid ratio of 60-20-20. Now I would like to compare this to the 4 systems that I summarized in the beginning. The iScreen, as said, is rather a screening tool with a high density. I did not find an exact number here. But also a high luminal short-chain fatty acid concentration in the system. And that's likely because there is no removal of metabolites. So the short-chain fatty acids would pile up and could eventually cause a change in microbial composition also. The short-chain fatty acid ratio also deviates a bit from that of humans. However, the adaptation time of microbiota in the system is short. In the ARCL system, we have a high bacterial density of 10 to the 7th. I wasn't able quite to calculate the short-chain fatty acid concentration because they use a throughput dialysis value that is published here, but the short-chain fatty acid ratio suggests it being physiological, so we could assume that also the concentration is physiological. The adaptation time takes 3 to 4 days. Moving to the chyme, we have an increased number of bacterial counts or density. I will end in half a minute. However, a low short-chain fatty acid concentration in the lumen, and that's perhaps, I hypothesize here, related to the lower bacterial density as such. However, short-chain fatty acid ratio is physiological. Adaptation time is rather long, as 14 days. I'm not sure here to what extent then the composition is rather adapted to the system or whether it's representative for the physiological situation. And the last one, the TIM system has 10 to the 10th bacterial density, 137 short-chain fatty acid concentration, and a short-chain fatty acid ratio that also resembles to the physiological one, as you can see, and about 16 hours to a day adaptation period. In my last slide, I would just highlight that it is hard to understand whether we are using or whether we are getting the right results from the in vitro system, simply because the data of proximal colon knowledge from humans isn't there so much. So we use the best what we can by using stools because it is easy accessible, and it is also not that far away from what is representative for the proximal colon, even though. And as I said in the beginning, it is important to see that we simulate parameters that are really representative for the in vivo situation, because otherwise the outcome isn't either. Thank you very much for your attention.
Okay, super. Thank you very much, Susann. I cannot see any questions upcoming, but I will step in with a question of myself. As we learned from Gwen, there are various physiological factors that affect the diversity of the microbiome and its functionality. So are there any systematic studies when you play around with transit time, quote unquote, which means technically, you know, how far and how fast you push, and how that then affects the digestion, the production of short-chain fatty acids in the TIM system, for example?
Yeah, sure. So I have to think a little bit what I answer. Of course, the transit time in humans varies, and what we see that the composition of the microbiota depends not so much of transit times, but also the substrate that we supply and the surrounding conditions as such. So in that sense, if we would alter the pH in the time course of 12 hours, saying traveling from the proximal to the distal colon, or if we would alter it shorter or longer than that, that will also impact on the microbiota as such. And the thing with in vitro systems is we can put any conditions to it, fast, slow transit times, or physiological or weird substrates, the microbiota will always react to it.
Okay, let's stop here. I saw there were some questions coming in and compliments for your presentation.
Thank you.
So we reconvene in the end in the panel discussion, and some of the questions will be brought back. So it's my pleasure now to introduce Professor Dirk Haller, a former colleague of mine. Yeah, Dirk is a chair of nutrition and immunology. He also is a director of the TIM Institute of Food and Health, and coordinates a huge center grant application sponsored by the German Research Foundation. So yeah, Dirk is doing research in humans, but also in gnotobiotic animals. And yeah, I'm looking really forward to your presentation, Dirk, to share your knowledge with us.
Thank you very much, Hani. It's a pleasure to be around. I try to move this thing. I don't know if the slides, if I can move the slides forward. It's still loading, I think.
There is a delay in the slides, but they are moving.
Okay, so I just talk about slides which you don't see. What you have, I think, what I have seen in the last two talks is that we start to learn much about the microbial ecosystem in the intestinal tract. And as you will see in my title of the talk, I think one of the key relevant questions is to understand the functional relationship of these bacteria with the host, their interaction with the host, and their role in certain pathological situations, because I think that's where we speculate the most. And I have the task to talk a little bit of translating all of that into in vivo models, and you will see that having an in vivo model is also very complex, and you need to make decisions on what you want to answer in your question. But you need to learn something about your human setting before you want to address questions in the in vivo model. I have not much to declare, and it would be fantastic if this slide would move ahead. Any idea?
There is a lagging on the system, but try if it.
Here we are. Fantastic. So functional relevance. That's something, and I think the challenge is to translate observations in humans into model situations. I think one thing I want to repeat, based on what has already been said, is there is substantial variation in human populations. And you can see here two larger populations. One is the Chinese cohort, and one is a cohort which is published. And from the color code, you already see, that's the good news, is that the structure of these ecosystems are very much shared if we look at a higher taxonomic resolution. It's mostly dominated by firmicutes, but even at this phylum level, you see that there is substantial variation within the populations. And we're talking here in the Chinese cohort with more than 7,000 people, and this is a cohort of 2,500 people. And we already heard that understanding metadata and variables which could explain this variation is very important. And at the moment, I think we understand roughly 10 to 20% of the variables which would influence this inter-individual differences. So keeping this in mind, having said that there is probably a large variation within individuals and populations. And if you move from 16S profiling now to shotgun analysis, I think you improve complexity, but you also come closer to reality. And what you see here is basically a phylogenetic tree of 3 different species here from core microbiota, and the left hand here is a Faecalibacterium prausnitzii. And if you have the chance to look at strain level diversity, one realizes that there is massive individual variation, and all these data points are basically individuals of a phylogenetic diversity of different individuals. They cluster according to countries, for example. And at the end of the day, we have to assume that probably we are perfectly individual, having a very individual set of bacterial strains in our intestine. So one last point I want to mention is also, in addition to stool consistency and so on, it is the time of sampling. And what we looked here in a cohort of 2,500 in Cora and validated this in 2 other cohorts with a total of 4,500 people, is that you look at richness here, you look at diversity with the Shannon, and you look at the 2 dominant phyla, Bacteroidetes and Firmicutes. And in blue, it's the healthy, and in red, it's type 2 diabetic people. And what you see is a daytime-dependent oscillation under normal circumstances. And that fluctuation is actually, if you look at Firmicutes, it changes over the day within roughly 5% of total abundance. And that is quite a bit. And you see that this harmonic variation, this diurnal variation, is lost in type 2 diabetics, which would indicate that in a disease, these rhythms are disrupted. So sampling and knowing when you sample is very important as an additional variable. Then we extract signatures, and these signatures we try to link to a certain probability to develop a disease. And what you see here, when you look at sensitivity and specificity, and you can look at almost all of the microbial analysis at the moment, roughly 80%, 70 or 80% sensitivity is achieved in classifying diseased from non-diseased populations. For a risk profiling, that's okay. But if you would want to use this as a specific test in the clinic to say if I look at the type 2 diabetic patient or not, that's not sufficient. And the bad news is if you take your signature, which you developed in one cohort, and you move substantially to another one, you lose predictability. So regional resolution was also something which has been shown in this Chinese cohort to be very influential. So moving slowly to mouse models. This is a patient cohort. And again, I reiterate, we are very individual. And what you look at here is the beta diversity of a cohort of 35 patients sampled over different periods of times during their chronic inflammatory disease. The ellipses describe basically one patient and the fluctuation. So what you see is very, very individual configurations over time of a disease. And this is illustrated on the right-hand side. Again, a patient here displayed and his time course over time treated at point 1, and then he basically moves in his microbial configuration almost to the whole compositional field of all the patients. So during a disease, compositional changes are very, very dynamic. Okay, and if we now try to put this into systems, we have to be aware that if we work with a model system, we usually try to control the environment, the microbiota, and many things. But even in a normal housed mouse, you have from facility to facility differences, and you have differences even between cages of one rack. So at the end of the day, to control microbial ecosystems and to compare them, we a lot use germ-free models. This could be a mouse, this could be a rat, this could be later on pigs. It's a bit harder to work with because they're larger. And then you can colonize them and you control actually what you build in. But that has also a drawback, because a gnotobiotic or germ-free mouse is not necessarily physiologically completely similar to a colonized mouse. So again, that has basically consequences when you choose your model. And if you go to diseases, you need to choose your model based on the question you want to address in the disease. And again, there is no model explaining a complex disease like diabetes, like inflammatory bowel disease. But what you see here, actually, when we divide this cohort of chronic inflammatory bowel disease patients, red, they are inflamed, gray, they're in a period of remission. And we basically take now stool from those patients, transplant them in a mouse model of IBD, and you see actually, if you look at the tissue pathology, that you can transfer the disease with stool coming from a patient which is at the moment active in disease. If you do the same thing with a microbiota from a patient cohort which is not in a disease situation, you actually don't induce disease in the model. So there is the potential to transfer via stool transplantation in mice. You can mimic the disease phenotype. And again, and that's the right-handed figure here, if you transplant the stool of one individual, and these ellipses show this, there is basically the donor sample, and then there is basically the sample how it grows in the mice. They are close together, they are not similar, so you lose certain bacteria which do not grow in the mice. But nevertheless, what you see is that there is different configurations which lead to the same pathological endpoint. So once again, we're not talking about one configuration of microbes inducing a whole set of different patient outcomes. There is probably also very individual interactions of different microbial configurations which lead to the disease. And that makes it, unfortunately, a little bit more complicated to define general signatures. A last word to this mouse model, that's the last slide, honey, is if there is a blue line and there is red peaks. The blue line is basically is the input sample, and you see the OTUs, which is the molecular description of a species. So you have relative abundance of species, and you have more abundant and less abundant. If you colonize now a mouse, it's not that you completely transfer the same configuration from human into mouse. No, there is certain bacteria which spike out and grow much better in these mice. And there are then, relative to the outcome in the mouse, you have probably selection of bacteria which could be considered more as protective or aggressive. So again, there is brain needed in terms of interpretation of models. And unfortunately, it's a very selective scenario. So I just want to briefly say you can combine this with model systems where you bring all the data from mice and humans together, and then you apply mathematical modeling, and these ROC curves then give you sensitivity and specificity. And it's very clear, the more information you put in, the higher the resolution is. So if you only work with bacteria and OTUs, you get a certain level of predictability, but you increase this predictability if you include, for example, information on the metabolite milieu, and you integrate these data. So we're facing a complex field which always works with these profiling technologies, which everybody would say, it makes me nervous because this is huge data set, there's algorithms hard to understand. At the end of the day, I think all of those data need to stand their impact in models, and you need to understand why certain bacteria do certain things. In the last slide, potentially, you have summarized this up. So you have a repertoire of different situations where you can colonize mice with mouse microbiota, with single bacteria, with complex microbiota, but you also can use human material to colonize mice and select human-related bacteria to address questions in mouse models. That's possible, and all of that needs to be fed back then to the humans again, and need to be validated in cohorts. And I think that's the trick. And I guess this is not moving, so we just skip my conclusion slide and jump right into the discussion. Thanks. Here's the conclusion slide, yeah?
Okay.
So you can read it.
Dirk, thank you very much. That was heavy load, but compliments. You have produced some nice papers recently. Are there any questions coming in now regarding the presentation of Dirk? Okay, here is one. Thanks for your talk. I missed the advantage of the animal model compared to the non-animal models described in the previous presentations.
Yeah, that's a good question. You know, I try to avoid this classification that's good and that's bad, because at the end of the day, you ask different questions in different models, and none of them will reflect completely reality in the humans. And some of the very cellular molecular aspects are even not addressable in a mouse model. So if you, for example, can generate organoid cultures from a human or a mouse, you can really then zoom into cells and address in a much more specific way the cellular mechanisms. And that's the same if you use ex vivo fermenters. You can manipulate an ex vivo fermenter in a much different way as you would be able to do this in a mouse. Nevertheless, a fermenter lacks the physiological environment. So again, I would not try to argue that there is good and bad models. I think there is a good and bad scientist who uses the models in good or bad ways. And there is good and bad interpretations. And that's, again, boils back down to the scientists and their use and their questions and their conclusions.
Thank you, Dirk. I guess a number of questions will be coming back in the final panel discussion. In particular, what I like very much that you addressed also this geographical variation. So keep that in mind, that we will talk about that later on. It's my pleasure now to introduce Dr. Michela Miani from the ILSI office in Brussels, and she's going to introduce an ILSI Europe new activity that relates to polyphenols and the microbiota, and later we have then Professor Kieran Tuohy coming in, and he already has done a lot of research on the same subject. So, Michela, thank you very much in helping us organizing the session, and the floor is yours now. We cannot hear you. I cannot hear you.
Can you hear me now?
Yeah.
Sorry. System, live problems. Thank you, Anna-Laure. Oh, it's almost tea time. How good would be a good cup of tea now? In ILSI Europe, we have several task forces and activities around the gut microbiome. And today I would like to talk to you about one activity in particular that focuses on the polyphenols and the gut microbiota that is tackled by the Health Benefits Assessment of Food Task Force. But polyphenols, so what are polyphenols? I have a lag in my system, of course. So polyphenols are micronutrients that we can ingest through certain plant-based foods. And for instance, I think that we are gonna eat them much more than we think we would. So if this was your breakfast this morning, where would you think to find polyphenols? Well, it turns out that you would find them in many of the items, in apples, in the juice, in the jam, oranges, and even coffee. And actually many more, from berries to chocolate, from red wine to the tea that I mentioned in the introduction at the first slide. Polyphenols are usually associated with health benefits, from cardiovascular to improved gut health. But actually, the mechanisms for these effects are not fully understood yet. And gut microbiome composition is modulated by polyphenols. And there are more and more evidence that say that polyphenols can modulate the gut microbiome by contributing to the maintenance of the balance in the microbial ecosystem. But how and which polyphenols or which metabolites, this is something that we will try to answer during our activity. How we're going to do that? So we decided to take the approach of a systematic review. And throughout the activity, we will be disseminating our findings and some interesting fact about polyphenols. So if you missed the two first episodes of the vlog, please find them on social media. And of course, we will be deciding where and how to better disseminate our activity through conferences or maybe webinars, and of course, an open access publication. So then what? What is going to be the outcome and the impact of this activity? So first, it will be to understand the link and how polyphenols and gut microbiome interact. And then secondly, identification of potential polyphenols that could be targeted for health intervention. And finally, support health application for polyphenols. Health benefits and health claims are at the core of our task force. So if you need more information, if you want to know more about what we do, please visit our e-booth. Thank you. Hannelore, hey, hello.
Here I am. Thank you very much. That was a nice intro into this activity of ILSI. And you have so colorful fruits in the bag. There's a good symbol for all these colorful chemical structures we talk about with the polyphenols.
Indeed.
1,000 different molecules that in essence can be exposed to the microbiome. And there are only a very few good examples how important the microbiota is in mediating some of the beneficial effects of the polyphenols. So are there any questions to Michela? I can't, hold on a second. No, I can't see that. Michela, how long is this activity lasting then? Yeah.
The activity was just kicked off in April this year, so we're at the beginning, and we will think to be, completed within 2 years. But systematic review can be challenging to undertake, so we have many things ahead, but the group is really, really committed, and I think that I don't know, we are already progressing very well together. And as I said, if you're interested in the vlogs, they're on LinkedIn and on Twitter. And so far we have 2 out, and the next one to come at the end of the year.
Thank you. I think that's a very important activity under the umbrella of ILSI. So let's stop here in terms of the presentations and bring back all the speakers to the final panel discussion. We have almost 28 minutes left, and I'm almost sure that there will be many questions coming in. And now, for the first time, we also see Kieran. Hi, Kieran.
Hello.
Kieran is a professor at the Fondazione Edmund Mach in Italy, and has been involved in a lot of research activities around polyphenols and microbiota. And he joins me in terms of moderating, asking questions or answering questions. So yeah, all are back. There's Gwen, there's Susann, there's Dirk, Kieran and me. Super. So we have time for the questions. I haven't seen the data from our little poll, Michela. Yeah, have you any information on what our participants are doing in terms of microbiota research?
So can you see the results now? Let me check.
No. No, I can't see them.
Here it says that I shared them. I will solve this technical problem.
Okay, so let's start the discussion then. We have a first question. Unfortunately, I can't identify the person that is asking that. But here's the question to Dirk and Gwen and Susann. What are your thoughts on big companies which are becoming very popular, which are making bold claims about providing accurate profiling of the microbiome and specific tailored dietary advice? So,
well,
Gwen?
maybe I start on that because I showed data on this, and I think it's do you hear me?
Yeah.
Okay, so I think it's pretty clear that we can identify risk profiles which give a certain probability of classifying a certain disease from a non-disease. It's becoming clear that you maybe even use this as a predictive tool, but the accuracy, and I think that's the misleading point in quite a bit of the interpretations. The accuracy is still not good enough that a clinician would take it and say, okay, I make now a decision based on this analysis to go in this or the other direction of a treatment, for example. And if you look at calprotectin, for example, as a marker for inflammation in stool, which reaches above 90% accuracy in these classification analysis, and still the clinicians would say 90% may not work for the patients. So I think we're a little bit too bold in the interpretation and create too much. People expect too much from these analyses, and I think some companies dramatically misuse this because they attach claims to it, like with this profile you get diabetes, with this profile you get neurodegenerative disease, and I think that is even not only bad science, but also I would say borderline possible to do this. So I'm pretty clear on this.
Yeah. So you're touching upon ethical concerns. And would you like to comment on that as well, that the commercial world provides some claims associated with microbiome profiling that are not enough substantiated?
First of all, I need to say that when I look at these data, when I get them, and I get them a lot, because people first go to one of these providers and then they contact me, and then I get these reports in my mailbox of worried people that think that they are ill because they got a result from one of these companies. I need to say that technically, most of these analyses, they seem okay, right? I think what they're doing and the sequencing work, that it seems more or less aligned with what we are doing. But then there are the conclusions that they're drawing from that, and they prefer, many of them prefer giving their results as a list of genera or species even, and they present it like blood analysis. So there is a threshold next to it, and next to the threshold is a red flag or a green flag, whether you are in line or not in line with what you're supposed to have in your gut, in your feces. And I mean, I've been looking at metagenomes and or 16S profiles of individuals for a very long time, and based on the relative proportions that they're presenting me, I would not be able to draw any conclusions, but they're doing it, right? They're getting people worried, and then some of these companies go further and are providing advice that has really no scientific background. So that's one point, and the other point is, if you're contacting this company and you get your stool sample analysis and your stool samples analyzed, I would definitely check what they're going to do with your data and where it's going to end up. Yeah, because you're also providing a lot of material to a company, and you don't know what they're going to do with it afterwards. So that is your concern as a customer, and you need to be careful with the things that you're sending in.
Thank you very much for making this point, Gwen. I think that is a very, very valid point. So we have more questions coming in, and one is in particular also including then Kieran in terms of EFSA and EFSA claims around probiotics and prebiotics and symbiotics, etc., etc. So how far are we from such claims that are really substantiated?
First of all, I'd like to thank all the speakers, and please feel free to jump in at any stage. This question about EFSA is specifically addressed on prebiotics, so it probably overcomes the safety issues or the product characterization issues that they had with some of the other functional foods like probiotics. But prebiotics, I think the challenge is really proving cause and effect with a recognized biomarker of health. So something like changing levels of cholesterol, and we need more studies showing cause and effect with ingestion of particular prebiotics with health claims that EFSA recognize as a real improvement in health. Of course, the other angle there is we can show that prebiotics change the balance of bacteria, the relative abundance of bacteria in the microbiota, and is that a health effect? And that kind of goes to the last discussion as well about the big companies providing profiles of bacterial names and claiming a health effect with these profiles. And maybe something that came in from the other speakers as well. Is it time to move away from bacterial taxonomy and more to what the bacteria are actually doing in terms of metabolites, be it butyrate or other short-chain fatty acids, or the small phenolic acids from polyphenols? What is the actual mechanism of effect of prebiotics, but also of gut microbiota in terms of human health? And until we get a good grasp of that mechanism, I doubt we'll be able to design properly powered human studies to prove cause and effect between microbiome modulation and recognized health parameters.
Okay, thank you very much for this clear statement on that.
I have the results of the poll. Yes.
Yeah.
So I don't know how to share it, but I can read them. So in the attendees, we have 13.3% of people that are interested in vitro and 30% in vivo. Both is major, so it's 46.7, and other techniques 10%. There is another poll that we would like for you to answer is, I'm gonna try, so let me see. Oh my, open poll. Okay, now you can see it. Currently, only about 10-20% of the variance in the human microbiome can be explained. Do you think you can reach 80%?
Do you think we can reach 80%?
Well, if you look at human sequencing data, I think that's the blueprint of what we can expect from the microbiome. So at the moment, if you look at GWAS data explaining heritability, you roughly reach a threshold of usually 20 to 30%, and the rest. So this gap is probably minor contributions or combinations of variant genes in the human genome. So they add up, but the effect size is probably relatively low. And looking at where we stand at the moment with variables concerning the human microbiome, we probably already picked up on very dramatic variables. I mean, if you look at the Chinese cohort, which I think nicely showed that there is a regional effect, a substantial regional effect, explaining 10% of variation in the data pool. And then you have plenty of effects of the physiology, and not always disease is number one in influencing. So I think we picked already quite good repertoire of these variables. I think the question is not if we can identify at the end all variables which explain all variation, because we are all probably very different. I think the trick is to understand core mechanisms of how bacterial networks function as an ecosystem and then translate their impact on the host. And for that, you will probably end up with suggestions which explain, I hope at some point, 60% of what we see in terms of endpoints. But looking at the GWAS world, we're now facing 20 years post first genome sequence. Well, I guess we have to have a long breath.
Yeah. So, Gwen, would you like to comment on that? Dirk was optimistic in saying that 60% of the variants he explained in some years to come.
First of all, I think Dirk was also saying that there is no need to be able to explain 80%. Is that correct? Yeah. So, and I really agree with that point. It's I think we can, if there are some fundamental things that we can be able to explain, we will be able to exploit the power of the microbiota. There is no really a need to understand why this person has this particular species and the other person doesn't have. And the other thing, what remains really difficult in microbiome research, and that's part of the what we call the individual variation, is part of the food that people are eating. It's really difficult to control that and to have conclusions on the impact of the food on the microbiota, because the diet is different for everybody. The tools to collect data on diet are not always very simple, and the results are not always reliable. And then you also need to take into account the lag time between when you eat something and when it's actually fermented, and all these things make it very difficult to map the impact of the diet. So I think there, maybe with models we'll be able to. I'm thinking then of the in vitro models, but in vivo, in humans, it's very difficult to map that fraction.
Yeah.
Thank you very much, Gwen. Maybe a comment. You have produced a wonderful paper where you looked at all the effects of dietary constituents. And that shows that almost everything you put into your gut has an impact, and almost always the same impact in terms of the fiber and the total carbohydrate and sugar and alcohol, red wine, white wine, coffee, etc., etc. So yeah, that was a fantastic paper. So coming back to the issues that we were discussing in terms of in vitro versus in vivo, and there was a coming up to all the speakers. What about the organ on a chip model, pro and contra? How to increase the explained? Okay, so are there any comments on that, Susann?
Yeah, so my first thought would be that indeed it is good to combine cells with actual physical models like I presented, for example, because this is really something that is lacking at the moment. And on top of that, there are a number of other factors which some of the models don't take into account, like the mucus layer. And for example, the Scheim uses these mucus balls, which allow also the growth of mucus microbiota, which also play a role. So I do certainly think that this gut on a chip, which has a different approach than the models that were presented here, have an added value, and especially if it is integrated. So we are not there yet, but integration of the two would be definitely advantageous with regard to generating responses also on a cellular level. The organ on a chip, as far as I am informed, and if somebody is there in the audience who has more experience on this, please jump in. But these systems are at their very starting, at their beginning. So there are some sort of diffusion models which use simple epithelial cells, 2 layers, having an inside-outside infusion of colon material, and then looking at translocation back and forth. But there are also models that use differentiated stem cells, which resemble then colon tissue, so to say. But as I said, this is very premature, and this is at the beginning of being developed. But I do think it is interesting.
Okay, thank you. Maybe Dirk, you can comment. You have, and I have been doing some organoid work.
Well, we do extensive work on organoids, and again, I think it's really important to understand the limitations and to define your question you want to address. In our hands, the inside-out organoids are potentially a tool. If you work with normal organoids, you have very little luminal areas where you want or not want to inject stuff. It becomes difficult. You have the 2D cultures, which again are problematic in forming tight layers. So I think the organoids are fantastic to use molecules produced by bacteria, which then can be tested on their functionality in terms of cellular resolution of the epithelium. Where do they act? Do they act on the stem compartment, on goblet cells, Paneth cells? That's fantastic. If you work with bacteria, in our hands, that's already problematic. If you want to work with complex bacterial mixture, is close to impossible. And then I would say, what's the benefit to work with a complex microbiota when you have, on the other hand, the possibility to use gnotobiotic mouse models? So I think the choice of models be done in an intelligent way to complement the conclusions. And at the end, you need to move this back to the humans. So I think organoids are fantastic. It's a breakthrough in working with epithelial intestinal material, but they're not usable for all the questions.
Thank you very much, Dirk. I totally agree with what you said. Susann, comment?
I think the major advantage of those is that not only single cell layers are simulated, but all the different types of cells present in the gut, because those interact with each other, they impact each other, and so on. I think this is the major point of organoids versus flow-through systems, for example.
Yeah. So that's clearly an advantage over the classical KCO2s or HC29 cell models, no doubt. And I got a comment from Isabel from ILSI, that ILSI Europe contributed to a potential EU-funded project on organ on a chip. Fingers crossed that the project will be funded. Okay, so yeah, there are more questions coming in, and I take one from Henley Bryan. What is the role of change compared to randomness in the microbiome in gut and elsewhere? Is the microbiome a partly random process? So is this any just stochastic? Is a question to, I guess it's Gwen or Kieran.
Well, what I hear, I start with a possible answer. What is clear from literature is that under the umbrella of species, you can gain and lose strains. And sometimes it's possible that you put a strain in an ecosystem and the strain stays there because it fulfills a niche function. That's all good. But sometimes it's just a competitor and it's pushed out of the ecosystem. And I guess the random nature could partially come from the stability of strains in an ecosystem. The question is from where these strains come from. Some are extremely anaerobic, and I think that's the boiling questions which need to be addressed. But a certain level of, I would say, fluctuation of strains, even species, is absolutely visible in the data sets when you follow up longitudinal data. Yeah.
Okay, thank you, Dirk. Gwen, would you like to comment?
So yeah, I think it's if we look at how the microbiome in the human evolves from birth to how it matures, we see that there are a series of succession events that take place, and we indeed see that there are some niches that need to be occupied in these succession events in order to be able to reach this mature eubiotic configurations, right? But these niches, they can be occupied by several bacteria, several species, even several genera, and it doesn't necessarily need to be the same genus occupying this niche. And we see that the end result might be very different, but in terms of metabolite production and these things, they're still very much alike. So yes, there is definitely some randomness, but well, it's a randomness in a controlled setting, right?
Yeah. Thank you for this comment. There's another question coming in from Damien. What's about the in situ sampling of luminal contents in humans throughout smart capsules with compartments inside and similar systems? So remote controlled sampling where you have a little capsule passing down your intestine. Who can comment on that?
I can comment on that. So that's a very interesting question, and for both ways. So I think there are two major applications for this type of capsules. One is a sort of a diagnostic tool. We touched upon this earlier. What do we measure in our gut, and what does that mean for our diet, for example? That was the very first question. But on the other hand, These are really nice research tools, actually, which we, for example, used to validate some of our settings. So these capsules who you can swallow and that travel down the GI tract, they can nowadays measure pH, take samples, convert all kinds of data to databases outside the body while traveling through the gut. So I think for this application, it is really fancy to work with and cool. But I'm not so sure whether we are there yet, as discussed in the very first question about using this as a diagnostic tool.
Yeah, I totally agree. Super. Thank you. So I would like to come back to this geographical variation. And Dirk mentioned it in the Chinese cohort. I found it absolutely impressive. What they demonstrated, and what they also showed was then when they developed an algorithm to predict, for example, glycemic index responses, and used this model in a different region, it didn't work anymore. So I'm not aware of any study in Europe that, for example, compared the microbiome of the UK people in London with that in Newcastle or in Leuven as compared to Brussels. So how important is that in the total context? Dirk mentioned 10%. Would you agree, Gwen, that it is this range of variability that comes with the region?
I would like to comment on that. So I do think that perhaps not so much Brussels versus Amsterdam, but I do think it is very interesting to look at the microbiota of indigenous people, for example, who eat roots and skins of plants and whatsoever. And these people seem to have a completely different weight of microbiome massa as compared to us Europeans who hardly eat fibers and so on. So I do think that there are differences visible there. In TIM, many years ago, we studied, for example, microbiota originating from a fast food population originating from the US versus healthy middle European something. And yeah, of course, there are differences there, of course.
Yeah, but people that consume a large quantity of fibers also have completely different stool. Not only microbiome, it's also volume and water content and things like that.
Yeah, of course.
So coming back, is this an issue? Is regional variation important here? Yes or no?
Well, in the last study which we published on this rhythmic behavior of bacteria and the extraction of signatures, we had 3 cohorts, one in the southern part of Germany, one in the northern part, and one in London, UK. And the transferability of the signature to London, UK was not possible. Yet, I think the drawback is in that study, it was the DNA extracted was the DNA extraction took place in London, sequencing took place in London. But between the northern and southern part of the German study cohort, it was almost everything the same, not everything, and you could really see the cluster effect. So I think what is important is that stool samples of different regions in Europe need to be compared in the same platform, and then to nail this down, because the application, I think, is quite substantial, because then you cannot maintain a claim that a postulated risk signature is applicable for different cohorts away from where you have basically extracted this information. So I think it's important, and you need to look at this, and this is one of the variables which affect our outcome.
Kieran, would you comment on that? Because when we talk about Mediterranean diet versus Northern or Nordic diets, I mean, polyphenols are all over the place, but diets provide quite different patterns. And how good is the evidence that the microbiome, in context of this regional or geographical variation, affects the outcome?
I do think we get sidetracked a bit by the geography and miss the bigger picture, which might be the diet. And how different is the diet in the different countries? Is it realistic to expect a big difference in diet between North and South Germany? I don't know. Probably between the UK and Germany, you're getting closer. With China and Germany, you probably have a fairly big dietary diversity. But also, if we drill down into the drivers of dietary diversity, it's going to be fiber, polyphenols, live microorganisms in the food, fermented foods, and what exposure we have to fermented foods. So these factors, I think, should be brought into these discussions about geography. And it's not necessarily about the pin in the map, but more about the other metadata that go along with this idea of geography.
Well,
Maybe the strain thing, Dirk, do you think there's an issue at the strain level as well that might be more associated with individual populations? So passage of strains between mother and child or within communities, and isolated communities being probably more diverse than others.
Well, if you believe Nicolas Segata, which is, I think, the only one at the moment who really digs in this strain level diversity in a extensive detail, I would say yes, that will hit us hard. And I just have been with him on a video conference 2 days ago, and he said we are absolutely individual. So that again brings back function, because if we're absolutely individual and if having complete different repertoires of strains, and any microbiologist who works with different strains in the lab knows that different strains from the same species, they really do sometimes very different stuff. If that's a reality, we're facing very individual configurations, and we're at the moment focusing on metabolic configuration, that the bulk of bacteria do certain processes, and that's fair enough. But I think at the end, it's gonna be at the strain level, compositional differences of the cell wall, differences in their metabolic repertoire, maybe not huge, but maybe important. So yes, I think strains matter. But one thing I realized when we did the study in Munich is having information of time of sampling. You know, when you look at the differences of abundances, 5% variance, and if that's not recorded, you can basically go quite off board in terms of interpretation just because you sampled at different times.
I'm sorry, but yeah, thank you, Hannelore. Exactly, our time is up. I would like to thank you, Hannelore, for the excellent moderation, all the speakers. Just a little bit note, more than half of the people think that we will know more about the 80% on the microbiome in 5-10 years. So there is a lot of work to do. Optimistic, but, you know, optimistic and good faith.